Okay, so we're going to have now a short session with five talks of 15 minutes. They have time, so if they finish 10 minutes, you have 5 minutes for questions. And I think that's the rules. And we're starting late, 3 minutes late, so I won't cut you short. So you first, who's Paulo? Ah, yeah. Okay, Paulo.
Yeah, okay, thank you. Good afternoon to all. It will be not easy to make a presentation in 10 minutes about so vast subject, but I will try. Let's see. I start this presentation with this slide related with the Global Climate Risk Index. It was established by Eriksson 2020, based and supported by a publication, a German publication, that give us an idea about the most risk areas of the world in terms of effects of climatic changes in the countries and areas of the world. As we see, the Asia region and also in Europe, some countries have high risk values, for example, Portugal, France, and Germany. And this is a red flag in relation to the vulnerability of our systems concerning with, for example, storm events, floods, and so on. So it is very important to follow this kind of indexes, and it will be very important in future to follow also some variables or parameters that affect our vulnerability, as for example, sea level rise, and some other impacts of climatic changes. Let's see now. I give to here two figures. One of them of the IPCC 29 sea level rise with a more optimistic scenario or projection, optimistic RCP 2.6, which projects sea level until 2100 in 43 centimeters, and another worst projection with 0.84 centimeters, which means 84 centimeters. So it's not a good scenario, and in the more recent report of 2023 of the IPCC, these trends was not better. In relation also to the global surface temperature changes, as we know by media, it is at this moment at 1.5 more or less degrees above the tendency in the beginning of our century, but the tendency is not too low, is to increase until 2100. And if we stop now our emissions, we will go to need some centuries until to reach the values that occurred previous to industrial era. So, as a consequence of the global sea level rise, I have here in the left side of this slide, the left figure, we can see the relation between the sea level height and the recurrence frequencies of historical centennial extreme sea level events, as for example, storms. And what we can see? We can see if we have a storm with a return period of 1 century until the past recent, in next future, this kind of storm pass to occur 1 time per year. And this was provoked by the global mean sea level rise. So it's not a good scenario. And we see now in several parts of the world that the locations where the historical centennial extreme sea level rise already are occurring yearly. And the right side of this slide, we can see Europe, and the most dark colors indicate the regions where we can see now storm events that in the past occurred 1 time per century, now are occurring yearly. And in the future, the white circles of North Europe will go have the same kind of situation. So, as we see, in the aim of the climatic changes, we have sea level rise, we have the increase of intensity of frequency of extreme events, mainly in the high latitudes of the world, and the contribution of these main aspects to the coastal erosion are related with one aspect that I would like to highlight. When the sea level rise in the 1 centimeter, according with the name the Brund rule that was established last century, the lower lying sandy shores rated 50 to 100 times height this value. So if sea level increase 1 centimeter, we have a horizontal projections of this 1 centimeter in 10 centimeters, 1 meter, okay? So the beach stay over there, but it migrates to inland for that kind of value. I can also highlight that in Portugal, for example, during the last 50 years, the coastline is the most in the areas with low-lying shore beaches. The coastal retreat also reach 100 meters in some places. So we have the importance of relative sea level, storm surges, and the in southwest of Asia, the monsoons, as the main contributors to increase erosion rates. And what can we do? We can contribute to know the impacts of these kinds of variables in our system, and we have nowadays some recourses to monitor the variables concerned to coastal erosion, that is the topic of this presentation. So we have satellite-based platforms, aerial-based platforms, drones, and land-based platforms, video cameras. In the aim of our study in Aveiro University, we use now video cameras because they cover a vast range of temporal and spatial scales from 1 centimeter until 1 kilometer, in relation to space, and in time from seconds to decades. So we can have a vast morphodynamic issues addressed by this kind of data acquisition method. So, since we have data, we need to process this kind of data in the automatic fashion. And we have available now to us the artificial intelligence. And I can here define two approaches to process image data that provide, for example, for satellites, video cameras, drones, and so on. The machine learning is a subject of artificial intelligence, which use algorithms that learn from data to make predictions. And deep learning use the same principles of machine learning, but make a more detailed analysis of the data and can go deeper in that kind of analysis because it's a more complex structure and can be very valuable, for example, for image recognition. So here is a chart with several steps to fulfill machine learning study for whatever we want. I will go to detail here briefly. All of these steps from project setup until modeling in relation to one case study that we have in Portugal. So we have here, we have here several seed studies, and I will go to focus here in the Ericeira beach, in which we have an automatic wave overtopping coastal difference structure. And our intention is to analyze and to identify whenever this groin is overtopped by waves. So the project setup is the wave overtopping automatic detected. And the morphological approach is to use deep learning techniques, in particular convolutional neuronal networks, to detect in the automatic fashion these overtoppings. So data collection, it was established between June 2021 until 2023, several 4,000 videos. And the next step is to cleaning, to create a mask, to eliminate uninteresting aspects of the image, and to focus in the overtop, which is the white color that we see in left part of the image, left side. So, follow step is to analyze a vast sequence of images and in several weather conditions to give the opportunity to the algorithm to identify the contrast between the overwash or the overtopic and the ground colors of the images. So we train the algorithm with several hundreds of overtopping image sequences with cloudy, clouding sunrise, cloudy evening, midday sunrise, and so on, with overtopping and another sequence with no overtopping. So the algorithm was trained, and the follow step is data preparation. We trained the machine learning technique with 90% of previous created videos and reserving the last 10% of the videos for validation. The follow step is modeling. names hyperparameter tuning, in which we increase or decrease the layers, give more dimension to some nodes or decrease the nodes, and there are algorithms that do it in the automatic fashion. And to finish, the results that we find, I highlight here. Until now, we obtain an accuracy of almost 89% in the automatic detection correctly the overtopping, and when overtopping not occur, the model occurrence reach 90.7%. Conclusions. Machine learning techniques are promising tools for automatic detection of beach morphodynamics, as well as overtopping or another subjects, for example, recurrent detections. These techniques can form a work basis for implementation of operational automatic real-time detection service associated to cost risk assessment, but can also be used to predict short-term prediction, for example. And the main advantage of these techniques in relation to the conventional techniques is that they are replicable for the most diverse geomorphologic contexts and are not specific. Okay, it's fine. Thank you.
Good afternoon, everyone. My name is Camilla Sguotti, and I'm a Marie Curie Fellow at the University of Padova. And today I will present a bit the results of our workshop ReMSES that was funded by EuroMarine, which is titled Resilience of Marine Socio-Ecological System. So to start, why do we care about resilience? Well, we heard a lot about tipping points already today in the talk from David. And basically, what the last global tipping point report highlighted is that there are 5 major tipping systems that are on the brink of tipping point. And also that already 25 parts of the Earth system, including the biome, tipping points already occurred there due to external pressures. So obviously, tipping points are a hot topic at the moment, and they are tightly linked with this idea of resilience, because in ecology, resilience is defined as the capability of a system to maintain the same structure and function, even if impacted by multiple pressure. So basically, it's the capability of the system to avoid the tipping point. And if we look at the number of publications that were published with the word resilience in title or abstract, we can see that recently the number spiked a lot, probably due to this recognition of the presence of tipping points in the Earth system. And as I said, resilience is really linked with this idea of tipping points or regime shift. And just to briefly give you an idea how we can visualize resilience. So if we have a stressor on the x-axis and we have the state of our ecosystem on the y-axis, so the ball will be our system. So at the beginning, when the stressor impacts the system will be able to maintain the same condition because it is resilient. But as the stressor increases, the system will lose resilience, and at some point we reach a tipping point and will switch into a complete new configuration, like a coral reef will become an algae bed. And now, if I try to revert the stressor, actually the system is resilient because it's maintained by new feedback, so I will not be able really to go back to the previous state or just very delayed. So from a definition point of view and also a visualization point of view, resilience is quite an easy concept to grasp, but actually it's a very blurry concept, and there are some limits on it. So first of all, there are not really agreed definition of resilience, especially between different disciplines. Second, there are multiple methodologies that measure different things, and it's difficult to reconcile them. And finally, there is no real operationalization in management, especially in Europe. And this is why we had the idea to do this workshop, ReMSES, to try to explore and try to reply these limits of the resilience concept. So the workshop was titled Resilience of Marine Social Ecological System, from a mechanistic understanding to an integration to policies. So basically, if we look at the keywords, resilience was, of course, our main actor. Then we wanted to look at social ecological systems. So we wanted multiple perspectives, also from multiple, from research investigating different things. We wanted to understand more about mechanism as well. So what are the mechanisms that build the resilience? And finally, how to integrate this concept into policies. And we did this workshop in Chioggia in November, and we were 24 scientists from 6 countries all over Europe. There were a lot of early career scientists, and we were modelers, but also social scientists and experimental scientists. So the first thing we tried to do was we tried to resolve the problem of the definition, but actually we saw that was impossible to solve it, so we agreed to disagree. Because resilience is actually an evolving concept that is transforming a lot over time. And for instance, in ecology, it's tightly linked with this idea of regime shift and tipping points, but in social science, it's more linked with this idea of adaptation and transformation, so quite different. However, we thought that to actually be able to operationalize resilience, we need to mainly answer 3 questions when we look at a system. So resilience of which system we want to analyze, resilience to what stressors of the system, and then for whom. So are we measuring resilience for the people or for the system itself? So this is to give a better idea of what we are investigating. Then we moved into the methods, and here basically what we did was that we present different methods, and I will present you one later, and we tried to categorize them to understand how they estimate resilience. So basically we find out that there are mainly 3 categories of methods that estimate resilience in ecology, but also social science. So method that estimates attributes of resilience, so more like network analysis, trait-based indicator. Method that estimate resilience of a system, like early warning signal, integrated resilience assessment, or CASPRA, that I will show you later. And methods that investigate mechanism of resilience, like food web model, ecosystem models. And these are a bit less used actually still for reply the resilience problem in ecology. So I will now show you one method, the CASPRA, to estimate resilience, to show you also a bit how we worked. So basically what we did was that we had some case studies, and these case studies help us to discuss about the limitation of the concept of resilience and how we could advance a bit the field and also understand how to operationalize it into management. So the example that I will show you is basically the estimation of resilience of the Northern Adriatic Sea community. So we are actually close to here, so in Chioggia, which is in the northeast of Italy. And you can see the area, it's in the red square. And basically what we did is that we collected data on the commercial species community from the Chioggia Harbor. And so what we did at first was that we wanted to see how this community changed over time. So we simply did a PCA, a principal component analysis, and we actually find out that basically there were 4 main phases of the community, one changing in 1987, one in 1998, and one in 2012. And the interesting thing is that some of these change points correspond also to larger scale regime shift that occur in the North Sea and in the Baltic Sea. And then basically we looked at how the community was characterized, and we saw that at the beginning there were like more slow growing species, and at the end there are new and more fast occurring species that are becoming dominant in the community. So to estimate the resilience, basically we extract the principal component of scores of the analysis as a kind of index of the community, and I will just show you here the result for one species, so obviously there are more dimensions of it. And what we did was that we use a model, which is called the stochastic CAS model, that is not much used in ecology. And basically this model comes from catastrophe theory and is able to model regime shift of a state variable, in our case the community, depending on 2 control drivers. So we chose as control driver fishing because this area is highly exploited, and temperature because it's also an hotspot of climate change. And how does the model work? So basically, the community, our state variable, is controlled in the dimension by fishing. So if fishing is low, the community is high, and I don't know if you can see this, but you can see the community will be in the ice shield, and if fishing is high, the community will be low. And temperature instead control the type of relationship between fishing and the community. So if temperature is low, the relationship will be linear, and then as it increases, it will become discontinuous with the typical regime shift curve. So this model is able to model a regime shift with a cusp shape and depending on shoe drivers. And we can collapse the model from this 3D to the 2D representation. And basically this area here, so the gray area, this triangle, is the area below the fold, so the area where the tipping points occurred. So this could be thought as the limit of the tipping point created by the shoe drivers. So what happens is that these are the dots of our system. So the farther our system is from this gray area, the more resilient it is because it's far away from the tipping point. And the closer, or if it is inside, it means the tipping points occur, so the resilience will be low. So we use this model to try to understand the resilience of the Adriatic Sea community, and I will just briefly show you the result. So basically, this is now on the x-axis fishing capacity, that is the driver we use, and temperature on the y-axis. And this area here is the same area of before, so the area below the fold, and the dots represent the community. And basically what we find out is that when fishing was high, the community was not very resilient. But as fishing declined and temperature increased, the community moved out of this tipping point area and is now in a more resilient state, but clearly very different from what it was before. So basically what we learned with this case study was that the Northern Adriatic Sea community depends on fishing and temperature, that the tipping point was crossed due to the decline of fishing and the temperature increase. And the new community might look resilient now, but the tipping point is reversible, so we will not really be able to go back to the previous state. So when we look at this case study, I mean, yeah, nice, okay, we managed to measure resilience, but what we realized from the workshop is actually we need to combine multiple methods, because now we know this part of the system is resilient, but we don't know what mechanism brought the tipping points, and also what characteristic of the community created this resilience to the stressor. So we use basically this idea of case study to try to understand and reply some major problems of the resilience concept. And so basically we use all this case study and our discussion to draw some lessons learned that could help to operationalize resilience in management. So from a methodological point of view, we realize that it's important to integrate a different type of methods. That it's a bit what I said before. So we need to have the knowledge, especially on the mechanism, because it's there that management could actually probably have an impact. From a conceptual point of view, we need to acknowledge that resilience is not necessarily a positive idea, because if we are stuck in an undesirable state because we are resilient, this will actually be negative for the society. And also we need to realize that we might not be able to go back to the past system state. So if we cross a tipping point, we need to transform into something new. And from a general point of view, we find out that to adopt an operational definition of resilience, we might need to answer this pre-question that I showed at the beginning, and that we need to realize that resilience is more a moving target than something really that we can grasp. And so basically to include it to management, this will require a change of societal and also policy thinking. And this is a flavor of the workshop, and thank you for the attention.
We have time for a few questions. Is there any room here? Back.
And my question is related to the customer that we found. We line and not that the time that we domain of attraction. Like location will flip from one side to the other. And I didn't understand that the dot stay on one of the border. Didn't capture how you compute this from the equation. You said that it was a stochastic model, but I mean, your lesson is there.
Yeah, yes. So basically, I mean, maybe I can show you better later the model, but basically it's computed from the data. So basically you insert in the equation, like you have a linear model that put your drivers into the, and transform it into factors that goes in the.
Is data driven?
Yes, it's data driven. Yeah, so it depends on your past data, in this case of fishing and temperature and the community. And yes, obviously it's very difficult to use it to predict. You can say something about the past, and yeah. So this is.
Any other questions? Yeah. I'd like to ask you a hard question.
Okay.
How do you know it's a tipping point? Do you know, or do you?
You mean in the model?
In the model.
I mean, in theory, like the model computed so that when you put your past data of so you have your biomass and temperature and fishing, it will find the combination of the tipping points when the system switch from low to high, basically.
But it could go back again. No.
No, because the shape of the equation is this cascade bifurcation. So you have a bifurcation, and when the system enters the bifurcation, depending on this driver, it will detect the tipping point. I don't know if I am clear enough.
It's a bit like regime shifts. You know, you go from one regime to the next, but sometimes they go back. I don't know.
Yes, I mean, in principle, it depends a bit on the definition again. I mean, in principle, they could go back, but I mean, generally, it depends what definition you use. So we use the definition that when the regime shift occurs, they are irreversible. So it's not something you can revert, or maybe just in a very delayed time, but not like phase shift or.
Yeah, I know it's an impossible question. Nobody. So.
But it's important.
Yes.
Yes.
Great.
Okay.
Thank you very much.
You.
Yeah.
Sitting and presenting, that's an interesting concept. I'll try it. So how do I get this away? What is one outline? Hide floating meeting harmony.
Yes,
exactly. There we are. Good. Good afternoon. My name is Klemens Eriksson. Can I start? Yes. I'm from the University of Groningen, from the group in marine ecology. And I'm going to talk about listening to fish and marine mammals. I do this together with my postdoc. See if I can. Here she is. That's Annabelle Koch and my PhD, Marianne Watson. And in the background here, we can see an illustration of the beautiful Wadden Sea where we work. It's a barrier island complex in the northern Netherlands and Germany. But we're going to start in, if it works. Enter. Arrow. Click. Thank you. Thank you very much. We're going to start in the Baltic Sea. So I've been doing research with a number of colleagues in the Baltic Sea for the past 2 decades. And if we focus here on these red dots here, this is the eastern coast of Sweden. It's a very deep archipelago. And here we have signs of a regime shift. That's what you can see here, where the dominating predator in the system, perch, has decreased through the decades, while one of its main prey, the three-spined stickleback, has increased sharply. We started reporting on this about a decade ago, that sticklebacks were taking over all this base in the archipelago. And everything became very, very confusing because our fishery biology said, those who are doing the monitoring along the coast, they said, no, we don't know what you're talking about. We have perch everywhere. This was super confusing for us because where we were doing our research, it looked like this, right? It was completely full of sticklebacks. They dominated the system completely. So to sort of understand this discrepancy, you really have to look at the data making up this time series. And of course, these lines, they are actually a lot of data points. So if we look at this graph here, every dot is one time and one bay. So it's describing one bay in this archipelago. And that's actually consisting of more than 12,000 fishing events. So it's a lot of data making up these lines. There's a lot of variation. And if we start really to look at what this is, we can see that this actually, so here we can see the ratio of a perch over stickleback. And on this side, we can see there's a lot of bays where sticklebacks dominate completely. There's a lot of bays where perch dominate completely. There's almost no bays in between where we have a mix of these. So we have studied this system for a while, and what we notice is that there's a predator-prey reversal here, right? So if we have sticklebacks dominating this community, they eat the babies of the perch. But if perch are dominating the community, they eat the sticklebacks. So you get one or the other. So that's typical sign of an alternative stable state, of course. So we've done a lot of research. We also see that this is the case, right? We have this in this in-between area here. We see that we get an alternative stable state dynamic in the system. And what is more interesting is that this is a very spatial thing. So if we start in outer archipelago, we can see that sticklebacks dominate. We move towards the inner archipelago at a certain point in space, it shifts. We have a threshold, and instead the communities become dominated by perch. And of course, we were working in the outer archipelago, and our fishery biology colleagues, they do the monitoring programs, preferably close to the biological stations on shore, right? So they were in the inner archipelago. What also is interesting, at least I think, is that we can see a shift through time, right? So time 1 here on this axis, showing this threshold where we have a shift. That's around 1970, 1980, and it looks like this. And only really the outer bays were dominated by sticklebacks. This shifted in the 90s, so that somewhere in the middle of the archipelago, this shifts. And then it moved, and we could see that bay after bay flips from the outer towards the inner archipelago through time. So this shows how complicated it is with ecological regime shifts or tipping points. We really need high resolution data in space if you want to see this along a coast, and we need it in time. And this is a problem for us. So this is where I work today, in the Wadden Sea. Here is Hanna. She's been fishing constantly for 3 years. Every second week she's been out. And it's hard work, right? This is salt marshes in the Wadden Sea. And you can do maximum 3 nets per tide. That's what you can do. So in 3 years, she has been fishing like a mad woman, and we're nowhere even near getting a spatial resolution in the data that we can analyze any sort of complex dynamics. Oh, that's my cue to stop, right? So it is not very functional. If we need this detailed data, and we're going to use tipping points in monitoring or in nature conservation, we need much better data on mobile species. So that's where my suggestion comes in, to use passive acoustic monitoring. And this is Marianne, my other PhD, working with listening to the soundscape underwater, right? So why sound? Well, it's a non-invasive method. We don't need any ethical permits. We don't have to kill our fish. It records continuously, so it's not just a snapshot in time, but we can record over time and see changes over time. And it also indicates behavior. So if we are able to sort of connect certain behaviors to certain sounds, we can also see what the fish, for example, is doing. That's also the drawback of the technique, right? If they're not vocalizing, we can't hear them. So it's usually we only sort of record activity. So the first question we get usually is, why don't we just use cameras or fantastic underwater drones? They're really cool. You can drive around and look at things. And I thought I illustrate that with an example. So we are looking at artificial reefs outside Groningen. So Groningen is here. It's in the border to Germany. This is our beautiful sea. We have the ugliest, probably, coastline in all of Europe. It's a cement border or an asphalt border. And to enrich this, we have been starting to try to put down artificial reef structures, testing how they work, see if we can enrich the community. We did this last year, and we put cameras on this, and then we took continuous underwater video for 8 weeks. And these are the 2 best photos. And we didn't see any fish, right? So it's very difficult. We also took eDNA samples. And yes, we described the whole fish community in the Wadden Sea. This is a tidal system. The water comes back and it goes out. but it didn't give us any clue on how to evaluate differences between these reefs. But we also took hydrophone recordings. And this is the work of DiverSea. She listened through these 8 weeks of recordings and the controls, so 16 weeks of recordings. And she found about 6,000 different biological sounds. So that's a good start. 6,000 is much better than 0, I would say. 21 of those categories were likely produced by fish, and we could compare the differences on a small scale. About 50 meters from each other, we could see differences. Fish sounds usually look a bit like this. They are really evening persons. They vocalize at dusk. So most of the fish sounds are around 10 in the evening. They start around 8. So that's where we really hear fish. But there's also seasonal differences, and that we can see here. So here's an example. This is a drum vibration, and these kind of sounds, they dominated mainly in February, which was our first 2-week recordings, and they sounded mostly around 18. I'll see if I can hear it. So that's the sort of drum. It's really like a drum, right? That shifted. So in May, we instead got these types of knockings. A little later in the evening, around 9, it sounds like this. A bit like a boat, maybe, but it is a fish sound. 1 hour later, they disappeared, and instead we got this type of grunts. The grunts, they also dominated in June in this, and that's there we have to be a bit attentive because there's just one grunt. Sounds a little bit like a shotgun, right? I think I have one more. Yeah. So those are a bit more difficult to detect, right? But we could also see differences in time. So here are just from 24 hours of recordings. So I annotated all the different sounds we had for these 24 hours on the reefs and outside the reefs. And the difference is quite striking. And we could also see the predators. So those are the blue dots here. Something like typical seals, right? And they would visit these reefs at high tide. So here we can see how good it is to have continuous recordings, because you can really see the behavior of the seals coming into the reefs and I assume eating on all the fish there. So this is what we want to aim towards. So Janneke, she worked with us for half a year. She manually annotated 6,000 sounds. This is really hard work. Marianne has annotated at least as much, but you need to do this manually. What you would like to do is really to make automated sound detection. For that, you would use an image-based neural net. So I had the perfect introductions here. And what you would do is that you take this, you do it visually, and then you go through your sound recording with a sliding window, and it matches these pictures. But there is no such library, right? We can have all the AI techniques in the world, and this is actually not very difficult to load a neural network. It requires some skills, but I have on a bell, so I'm good. But there is just no library to go to. And we need that to be able to sort of match this to which fish it is. So we have some indications of some species, but we need help. And that is my sort of suggestion. I think we really need to start to coordinate our sounds, because there are many groups in Europe starting to work on fish sounds. So we are in discussion with FLIS, for example. I see some FLIS people, where we at least are trying to make a library of the types of sounds. So we at least get that right. And that's we can start to connect also when they figure out what a fish is, and we figure out what a fish is, we can share that information. And I think that's it's an important thing we need to develop, how we measure biology. If we're going to feed all these digital twins and get all this information to understand tipping points. I think my time is up.
And we have 3 minutes for questions.
All right.
You have a question?
I have a question. Have you thought about going to an aquarium to get all the different species kind of, analyzed?
Yeah, so it's not so easy because they always have a lot of background sounds. So we have recorded in many different aquariums.
Okay.
It's very difficult to find some kind of aquarium installation where you get a good signal and they do their natural behavior. Because if they don't do their natural behavior, they don't vocalize. So we, fortunately, in Groningen, we have these big stickleback arenas, so to say, where they actually, there's no pumps, and they also, they breathe and they create new generations in tanks. So there we can record them. So we have stickleback sounds. And then, of course, during the 8 weeks, we also fished every day. So we can see at least which groups are candidates for these sounds. But it's difficult to get the exact sound.
Yeah, thanks, interesting. Would it be an idea to combine with maybe active acoustic to like multi-beam type to detect multi-beam?
Yeah, so I'm not so good at that technique, but we have applied for funding from the next round of wind park projects in the North Sea to combine these two methods. It could work.
We should.
Yeah, I also.
Yeah, I was wondering, do you think it's feasible to work with low-cost technology in terms of, do you need very high technique in terms of hydrophones on quality? Because there are now ongoing, these low-cost hydrophones, but I know that, for example, frequency response is not obviously as good as the wood ones. So there could be potential limitation on using them.
Yeah, so I would have to ask Annabelle. I have to admit that, because she's the technical expert. But I think for fish is in the same frequency as ship sounds, right? Which is a problem. But that's a quite easy frequency to record. If you want whales, it's a different story. Then we need to sort of porpoise and that kind of sounds. We need more expensive ones.
Very interesting.
Hello, everyone. Can everyone hear me?
Yes.
Should I start?
Yeah.
Yeah, so my name is Glaucia Fragoso. I'm a researcher at NTNU, located in Trondheim in Norway. And today I'm going to talk a little bit about this project that we got funded from September. It started from September last year, which is called DiverSea. And the title of this project is Integrated Observation, Mapping, Monitoring, and Prediction for Functional Biodiversity of Coastal Seas. So we all know that marine biodiversity is declining and is declining in unprecedented rates. The reason for that is mostly because of human activities, and that comprises climate change, such as increasing sea surface temperature, also oceanification, which is the other CO2 problem. We also have problems with pollution, eutrophication, also plastics, microplastics, habitat destruction. Here in Norway, it has been a huge debate about deep sea mining, which is one example where another habitat, the benthic community, might suffer a lot in the Barents Sea because of deep sea mining. So we know that this is happening because of human exploitation, but also because of climate change, like sea ice loss. And also we have overexploitation of the fisheries resources. So we know that biodiversity is declining, but we still lack a lot of knowledge of how we can have like an overall view of the rate that the biodiversity is declining, because a lot of the research that we have is either like a few places, like the long-term stations that we have, where we have access to long-term data that is not that many, and we know that many places in the earth is not included. We also know that it's very local, so we see a decline happening in one coastal region, but then how can we transfer that knowledge? Do we see the same knowledge in another region if the methods that are used sometimes are not the same. So we need we have this problem that we need a lot of monitoring because we also don't know, for instance, we know the drivers, we know climate change is one driver and pollution is another driver, but we don't know how those drivers they act together in impacting the biodiversity. And we don't know also what is the implication that has for food webs and for climate regulation. So we need monitoring, as I mentioned, and this monitoring needs to be, as we have discussed by Britta's before, it needs to be also highly resolved, because if we want to know the trends in that we are losing diversity, we need to know also the small scales, right? To be able to know how plastic those chains are, to be able to see, to have an overview of the tipping points in the long term, not just in time, but also in scale, in space as well. So we need to have access to more data. We need to use more cost-effective monitoring where we have like highly resolved information, high throughput data that are affordable in many places. We also need to use those robots in places which are of difficult access, like the Arctic. Like if we have a sensor there, it's much cheaper than having a boat also going there all the time. And it needs to be affordable, so we're able to collect more and more data. So the DiverSea, the main objective of this project is to develop this integration of different approaches, mostly cost-effective, low-cost approaches, where we can use a different combination of data to try to have an overview of how we can actually analyze biodiversity. So once, like those technologies that we're talking about are like low-cost flow-through microscopes, like the plankton scope, eDNA, hyperspectral satellites, where we can have more information about the functional groups, like phytoplankton functional groups, low-cost technology like low-cost mini ROVs, where we can have a camera that can like map the benthic organisms. So it's a range of different approaches to analyze biodiversity, and also citizen science, where we can combine activities in the public where they can collect data for us, and then we can analyze later in the lab or in the computer. So the goal is to develop this integrative approach of looking to biodiversity using different methods, and also to combine all this data and try to interpret the data, which I think will be a second biggest challenge. So we would have to use a lot of AI machine learning to be able to understand, to kind of have a standardization that we mentioned so much today in previous talks. And in a way where we can, maybe the data might not be totally interoperable if they are not exactly the same method, but they need to complement each other. And we need to be very careful when we interpret all these different data together. So once we do this standardization, we would try to put that in a map to try to understand the trends of biodiversity changes within different drivers. And in the end, the latest objective would, like, once we can reduce the complexity of this data, we can provide like a biodiversity dashboard where we can show to the public, to stakeholders, what are we finding with our data, and that can help them for making decisions. So the DiverSea started last September, so we're quite new yet still. We are 19 organizations, and that includes universities, research organizations, business, and also non-profit organizations. We plan to have this for 4 years, and this is a call from Horizon Europe. And we want to, like, the main objectives is, like, the first one is to use molecular indicators. So we're gonna have this professor that's gonna be focused and in eDNA and also DNA marks to look into diversity of species. And we also plan to have all this data to be able to not just to understand to interpret, but also to reduce the complexity, as I mentioned, and also to derive essential biodiversity variables, which is also not a very easy task. It's still quite vague for many of us, but this is one of the objectives. We focus on the methods. So we're going to do that in many places in Europe. A lot of those places will be in Norway, from the south of Norway, like Oslo to Svalbard. And we aim to combine all this data and to integrate all this data to in the end, to provide an interactive interface for stakeholders, and also to boost transdisciplinary approaches between marine science and science-based information. So this is the main PIs of the project. So the coordination team is at NTNU, my university, and that's Professor Murat Ardalan. He's going to be focusing marine biogeochemistry. Glenn will be focused in molecular approaches, and Maria Asuncena Gonzalez, she's going to help us with the coordination. And within each package, working package, we have people with different expertise. So we have Professor João Souza from University of Porto, where he works a lot with marine robotics and adaptive sampling. So our idea is to put like sensors, biosensors that can collect data for eDNA analysis or collect samples for eDNA analysis. So we put them in those underwater robots, and he's very he has an expertise in autonomous sampling. So the robot they would track whatever we want to track. If you want to look into subsurface patchiness, they would recognize the chlorophyll concentration to a background, and then they would, the computer itself, the robot itself would target to sample that location through adaptive sampling. We have also Professor Irena Pavlova from Bulgaria, where she's going to be working with big data handling. Professor Robert Ryan is from NTNU as well. He's a statistician and biologist. Jean-Luc is going to be focusing in modeling part. And then we also have like a non-profit organization where it's Liga pra Natureza, that's like a non-profit organization, where Gonçalo Pristas will also be involved. So I'm a researcher. I'm going to be employed. I am starting next month my contract, and I'll be employed in this first work package, which is developing this, using these different approaches and sensors and methods and platforms to combine everything and to understand what are the patterns of biodiversity that we can find. So the methods that we have is spectral reflectance. We have them in a microscope, so we can have like a flow-through microscope where we can scan the spectral reflectance of organisms. So if we see like a phytoplankton, they will see like the shape of their color. So that can be used and can be aligned with hyperspectral satellites, like the PACE, which has been launched last week or a few days ago. But we also here at NTNU, we have our own mini satellites, hyperspectral satellites. So we want to combine the large scale and the small scale, both in space and time. We also have flow cytometers with imaging, like the CytoSense for plankton analysis. So that gives a lot of optical data and also imaging data. Flow through low-cost microscopes, like the plankton scope. eDNA is also another method that we have. And we also are interested in acoustics. We have a lot of active acoustics for fish and plankton biomass estimations. But also we have passive acoustics like hydrophones for mammals to recognize the communication of mammals. And we have a lot of robots as well. We have Norway is well connected with ferries. So we also have ferry box opportunities. So we aim to have like several cruises where we can use all these different approaches. And we have also other projects like Sailing for Science, where we have citizen scientists, people that are interested in sailing, and they can collect also data for us. The environmental biological variables, the biological variables that the essential biological variables that we are interested is mostly plankton, because that's mostly my focus, phytoplankton and zooplankton. But we also have people in our at NTNU that is also work with megafauna and marine mammals, and also fish, and also with benthic communities. One example of this combination of technologies that we have, like I mentioned in general, but we also have like here in Trondheim's Fjord, in Trondheim, that's mid of Norway. So we have a buoy that has a lot of technology, a lot of instruments, a lot of sensors. Those sensors that we have are like typical environmental sensors, like CTD, CDOM, DO, turbidity, nitrate. But we also have instruments that can take a look into pelagic communities. So we have the cytosub, which we can use for bacteria until larger phytoplankton. We also have a ciliate camera, which takes images of zooplankton, mesozooplankton. And we also have a UVP6, which looks into larger zooplankton. And also we have acoustic data to be able to see large zooplankton, some zooplankton, and also some fish as well. And we also have a hydrophone, not necessarily in the buoy because it makes a lot of noise, but a little bit also in Trondheim's Fjord, but in regions a little bit far from the buoy. And we aim to start sampling.
There's 1 minute left.
Yeah, this is my last slide. We aim to start sampling this spring, and we aim to combine all these different data to be able to understand the changes of plankton and biodiversity of plankton. So I'd like to thank you, everyone, and if you have any questions, just let me know.
Thank you. I don't think we have time for questions this time, but we can go on with the next speaker who is very in position. I'm going to talk to us about jellyfish.
Thank you very much for the invitation to present our working group, and on behalf of Isabella Dampa, who's joining online, and Andrea Ramsek, who's sitting in the audience. So throughout this talk, I'll talk about jellyfish and why they actually are regarded as a problem, and how we should actually challenge the current paradigms that are out there. And would like to discuss as well that we should include them into ongoing monitoring initiatives, and Marco Stello is the perfect person to convince that. Good, but if we talk about jellies, a lot of people are grouping two completely different phyla into one kind of guild, which are the true cnidarian jellyfish as well as conch jellies. And even though they share a lot of traits, have been around for quite some time, they go back, we have fossil records for more than 500 million years, so they have been around in our oceans in nearly unchanged form. They actually share a lot of key traits that allow them to kind of adjust to a lot of adverse environmental conditions, such as low oxygen concentrations. Both groups are characterized by a bloom and bust cyclicity. And we have heard about ocean currents. So these currents can accumulate these accumulations that occur during spawning into bays, and there lead to interaction with social activities, fishing, desalination plant clogging, which have raised a lot of attention, such as here featured in Nature in 2012, the attack of the blobs related to the giant Nomura of Japan that can actually attain up to 200 kilograms in wet weight. But basically, there is a lot of attention about jellyfish, and they're always associated with two very negative paradigms. And one of them is that they take over as we are stressing our oceans. And the other one is that if this is happening, we are actually in deep shit, because this is a dead end in the food web. But throughout this talk, I would like to challenge these paradigms. And first of all, it's very difficult to get long-term data series. And if you look at this global map, which is the most comprehensive compilation of data sets that are available, including gray literature, you see that there are some areas depicted here in red where you have a high likelihood or a high certainty of jellyfish population increases. But you also have a lot of areas where there is actually a low certainty or even a stable or decreasing population. Now I'm going to zoom into the data set that actually has the longest record. It's in the Bering Sea, and here you see a time series of 40 years. And basically, depending on where you snapshot this time series, you get everything. You get an increase, a decrease, or no change. So what does this actually mean? Jellyfish populations fluctuate. And the problem is that we do not know why. So a lot of studies are actually focusing on very few species, but what is generally driving, population dynamics of jellyfish in our oceans, we are still left with a big question mark. And the problem is that we are lacking a mechanistic understanding. What we know for sure, though, is that non-indigenous jellies are a problem. And this is now a drone image off the Israeli coast. And you see the extent and magnitude of these jellies that are actually blooming every summer. This species had lately also been highlighted as a species of key concern from the EU, but that's not the only species. We have a lot of non-indigenous species. Some of them are invasive, pose an ecosystem impact, such as Miniopsis that you see here from Danish waters. And if you look at this map, the red areas indicate where Miniopsis is present. And this is based on more than 12,000 presence-absence data sets or data points. But what is striking is that if you go back to Eric's very nice Wadden Sea area, there are hotspots where Miniopsis attains up to 1 individual per milk bottle, But in very close vicinity, they're absent or have only been confirmed by eDNA. So we had the hypothesis that actually ocean currents are driving these range expansion and distribution barriers of non-indigenous species. And what you see here is the stability of ocean currents. So basically in very warm colors, red is a very stable flow. It's like turning on a hose where the water current is only running in one direction. Whereas if you have purple, it's like a kid sitting in a bathtub, or jumping around where you basically have a net zero transport. And what you can see is that where there's a southward current along the UK coast, there's a very persistent flow connecting the English Channel, all the way up to Scandinavia. And we were fortunate that we actually had a natural experiment that allowed us to test the importance of ocean currents. So here in red dots, you see the distribution range of Miniopsis in panel A up to 2010. So basically widely distributed in northwestern Europe all the way up to northern Norway or mid-Norway. But then we had a period of very cold winters. And during this time period, Miniopsis was kind of contracted to the North Sea. But what is striking is that during one warm winter in 2014, Miniopsis colonized the entire range all the way to mid-Norway. And we were fortunate to have molecular data that we could then kind of link to the populations where they got extinct, such as in the Baltic Sea. And here you see the population structure before the cold sweep and after the cold sweep. And this clearly indicates that the presence of Miniopsis in the Baltic Sea after this cold period is due to a new genotype. So the old ones, they got extinct. So this clearly indicates that we have populations that can get extinct, but that there is a very swift recolonization from warm winter areas. And that ocean currents actually form a kind of invasion conveyor belt. So if you really want to manage, non-indigenous species, we need to keep these areas in mind. But how many species are actually arriving in an area? So we were then kind of curious to see where does Miniopsis come from and how do the populations connect? And these data now, you don't need to understand them, but it's based on 72 whole genomes that we sequenced and analyzed. And the take-home message of this is that when you look at the northern population, we could actually see that there is still ongoing gene flow and there are genotypes that are still introduced into the North Sea area. Whereas when one is looking into the Black Sea invasion that happened 40 years ago, this was a huge invasion, a large inoculum that happened 40 years ago, but there's basically no gene flow anymore. So the population is kind of isolated. Whereas when one is looking into the more recent Western Mediterranean range expansion, this is a clear signal of a secondary range expansion. So this is just to put up and highlight that if one really wants to prevent new introduction and non-indigenous species in general, then it's important to really manage these highly interconnected areas. So coming back to the jellification paradigm, do we see that jellies take over? This is still a big question mark, but non-indigenous ones are a problem. But what about the dead end? And here I would just like to present an example where we went to the Sargasso Sea and actually looked at the eel larvae, so European eels that are as endangered as tigers, you know, but still you can find them in fish shops in Denmark and Scandinavia. And the problem is that with eel, we have not closed the life cycle in the laboratory because we can't get the small larvae to feed. What we did is we looked at the gut content in the native habitat, and we found that more than 75% of the gut content comprised of hydrozoan jellyfish. So this is an indication that with molecular tools, you could actually now prove, you know, feeding interactions. But this is not the only study. If you look into the Mediterranean Sea, and this here is a bluefin tuna, a large proportion of the diet actually consists of salps and jellies. This is now based on stable isotope analysis. So I just kind of pulled two examples, but I think we really need to update and get a paradigm shift going that jellies are part of the food web and that they're an important food source for a lot of commercially important species as well. So the way forward, we need dedicated monitoring. And this should also include the early life stage. And here becomes very tricky because you have a small polyp that's sitting somewhere on the bentos, you know, and if you include new hard substrate, for example, in the Wadden Sea with offshore wind farms, you create a lot of problems, but no one's investigating that. And then we need to address the top-down control, you know, what are the mortality rates of the different life stages? And this is then needed to actually incorporate jellies into food web studies. To get there, and I think today's talks really highlighted that, we need to have an interdisciplinary approach and implement novel technologies. We need a refocused activity plan, and we also need to have a change of mindset, you know, from jellies that you just throw overboard, like Inga just said what they do with their monitoring data, to get a good time series going. And that is where our next jelly, or Jelly Next working group comes in, where we're not only drilling on a European network of jellyfish experts, but also our global network to actually come up with a guideline how we can actually move forward and include jellies into ecosystem models. So with that, I would like to thank you, and I'm happy if there are questions. You said 10 minutes, right? No one does. Yes.
Yeah, you mentioned this idea, you know, of different substrates and the difficulties with the polyps. I was wondering in citizen science to promote, you know, the people monitor with different substrates and create that kind of, you know, jelly hunters, but polyp hunters. I think this can provide a lot of data and very useful in some cases.
Yes, excellent idea. The only problem is that these polyps are 1 × 1 millimeter.
Yeah.
So it's very difficult to see, and you probably need to have very skilled people. What they did in Asia is that they actually had a camera system, you know, going underneath these floating docks. And thereby, with automated image recognition, they could, you know, estimate the polyp number. And you get amazing results. If you remove these pillars, you know, then in a fjord system, suddenly you have 0 jellyfish because you're removing all the asexual reproduction phase, right? But this is on a very local scale. So the problem is how to upscale, you know, the small scale interactions.
Yeah, although hard to hear related to the topic, first related to the topic of the science of it, because this is emerging from a working group in the Mission Atlantic, in EuroMarine, as funded. And I suppose that one of the benefits is the standardization of methods to
exactly. Yes.
So is this something that you plan to
Exactly.
Or
Yes.
something that
So very good point. So basically, it would be really cool to elongate this working group for another 2-year series, because what we are doing now is we had a workshop where we kind of, got all the cards on the table. What are the knowledge gaps and which way do we need forward to actually get some kind of a guideline? What should we prioritize and how should we monitor jellies in the future? Because it's very difficult if you're looking at something which is large and very sparse. You need to have large nets as well, because otherwise you have observation bias. So it's very, very difficult that you're talking about the same things and don't throw apples and pies in the same kind of pot. Yes, so that's the plan, to come up with guidelines that we can standardize this.
So jellies are intriguing, right? So I always wondered, there's not so many good sampling campaigns for jellies.
Yes.
So do you think we sampled them enough in the North Sea?
Very good question. I mean, locally, we have kind of a good effort, but the problem is that this is unanalyzed, So there is some fisheries institute that kind of collect them, but they're hiding it somewhere in the drawers. So Mark, that's where, an orchestrated effort to actually get all the data series on the table would also be super cool.
I think the question is, of course, if fishing gear is representing jellies very well, right?
But if you look at the time series of the 40 years, I mean, if you apply the same method over a very long period, then even though you are not catching 100% of the community, you still get an indication if you have increasing or decreasing trends.
Yeah, one more.
Because you're doing like this one, right? All data series are with jellies, it's super variable.
Yes.
So is that true, or is it an artifact of that we don't sample them very well? Because that would be a key, right, to get them into food web models to understand this information, and I find it very, very difficult. It's super interesting.
It's super interesting and it's super difficult, but I think if you have, like the DiverSea data set, where you have 40 years, where you have consistently used the same kind of method, I think this data set should be kind of, should be clean, so you can trust that there's this cyclicity. What is driving this, we don't know. But that's why I was also intrigued with your sticklebacks, because, we should talk more.
I have a very quick question, because we've only half a minute. The genotype changes is fascinating. So are they changing their genetics quickly, or where are they coming from, these new genotypes?
No, so the data I didn't show is that in the North Sea, during this recolonization event in 2014, there was zero genetic structure. We sampled animals in Belgium, the Netherlands, all the way to Norway and the Central Baltic, and there was zero population structure. So this indicates that there is, that currents are really mixing the entire population on a very short time scale.
It's fascinating. Yeah. No, thank you very much, and nice timing.
So, my name is Jaume Piera. I'm from the Institute of Marine Science in Barcelona. And first of all, I would like to thank all the previous speakers, because I think they quite facilitated a little bit my talk. I will introduce one project called Aneris, and the idea is going to this idea of operational marine biology. The acronym Aneris comes from, if you reverse the name, it's sirena, which in many languages is mermaid, and that's why we have the tail and the molecular surrounded there. So, just very short, as you can see in the yellow one of the key challenges for the ocean observing systems will be the facilitation, the transition from research to sustained integrated observations. I think most of the previous speakers has mentioned this, and we think this is a need, and particularly for the marine domain. So basically, the idea in Aneris, we are trying to create this idea of operational sensing like technologies for marine systems. What does it mean, this idea of operational? The idea is coming from the acquisition to finally interpretation and dissemination as fast as possible. And this requires many automatic, implicit systems. And we will try to do so. There are several technologies trying to cover this up, because obviously if we try to monitor this in the whole range of sizes, we cannot use a single technique. There is no single technique that covers, well, probably somehow genomics could be, but genomics needs a kind of a validation, and that requires, basically, we have been focusing on imaging biooptics and participatory. We have not mentioned explicitly acoustics, although it could be hidden somehow, in the participatory domain. The idea is to create these operational marine biology products by combining several of these techniques. You can see in the middle, so imagine that we could, combine different layers of different resources and then create these operational marine biology products. And as example, you can see there is one now large scale network on genomics is EMOBON. It's one of reference, but it's quite sparse in a sense that there are only few stations. I'm not wrong, I think there are 16 now working stations and 16 places all over Europe. It's really, really a small number. It will be difficult, to increase that, but we maybe combine with some, I would say, low-cost technologies, try to fill somehow, these special gaps. I give you some examples of combining different life technologies. The first part is imaging, and the other could be participatory. And you can see, for example, in some cases, we can have a picture of one fish with one ectoparasite. And for example, through these imaging techniques, automatic ones, the system could identify there are two different species, but maybe then we can send all the two pictures to a participatory system, observatory, trying to, to identify or to label such a kind of pictures is one of the first steps. And I will put in my talk a lot of, emphasis on this new citizen observatory, participatory one, because it's the one that we are developing. And I can give you some examples. This idea of this participatory, platform, we call it Minca. Minca is a Quechua name. The origin of Minca, it's a word of collective work for the community. And this is the idea, of the pre-Columbian tradition, to help each other in many, I would say, agriculture activities. This is still ongoing, the Minca or the Minga activity, particularly in Latin America, but we like this idea, of this collective, capacity. And I can show you an example how it works. For example, you can see, for example, some people can take a picture of an organism, and in many cases, and particularly in the marine domain, many people will say, no idea which is that animal or organism. And they can ask some help, and there will be that community that can help labeling, because there are some levels, different levels of expertise within the community that then they can help and they can provide the community identification. What we are doing now in Aneris, we are trying now to combine this with what we call hybrid intelligence. We are including now, or new now, artificial users, if you want, agents that they can be trained and they can help on the process, and I will show you how we plan to do that. There is also an extensive network of people involved in Aneris that could help, on covering these special needs that we have along the different sea beaches, European beaches. The idea is that we have several technologies that we want to end up with OMB data production, and we will have several case studies to validate this concept. And I give you some, for example, some examples. With this high temperature resolution on time series, we are using well advanced, instruments that we will go to this cable system. This comes from the EMSO. EMSO is a European research infrastructure, and in some cases they have cable-based systems. So that means that they could install these devices and sampling at very, very high rate. We are assuming that we will have from order to terabytes per week on information. So imagine the number of values we can retrieve, and there is no limitations in terms of power or even bandwidth, because the system is cable based. So there is no problem in terms of batteries or the number of pictures that you can collect on that. But I will focus a little bit more on this second case study, is the one on promoting this idea of larger scale marine participatory actions, what we call biomarathons particularly, which is the idea, engage as many people as possible and during a relative long period of time, all the summer season, summer from May to September. And we have tested this in a very particular area. We have tested this in the Catalan coast, in the north of Spain. And you can see that we consider to create what we call marine living labs. That means that whenever we consider this idea of citizen science, we do not only consider the participatory community and also the academic, but also other people, other communities that are really, really important. The one is the mobilizing community, the ones that are, really in close contact with the people, mobilizing them on going to the beach, offering facilities and so on. And sometimes the facilitating community that could be, the city councils, for example. The city councils, they can provide buildings in which we can discuss things, or buildings in which we can change our clothes and put, our swimming dress for going to the beach. But this is really important at the end. We have tested this in this past summer, and during 3 summers, you can see more or less what we have collected in this area. And now you can see in October, we have like more or less nearly 123,000 for observations with more than 2,400 species. This was in October. Now we have increased this more or less in about 25,000 extra. So imagine now we have like 150,000 in a very small area. It's probably one of the highest densities reported observations in a relatively small area. We are talking about 300 kilometers, more or less, of coastline, roughly, depending on how you estimate. But basically, this gives you an idea what potentially could be done if we extend this idea all along the coast. And this, just to give you also added value, imagine we can have many species of these 2,400 species. In many cases, we have thousands of pictures. And that means that we can create what is called, training sets for automatic identification systems. Because if you are familiar with these AI-based systems of the learning, with this range of thousands of observations already labeled, this gives us, opportunity to train those systems for automatical identification of those species. And this can, speed up the rate in which we can ingest all the pictures. Because one of the problems, is that if we are now collecting like 70,000 pictures per season, but we double or triple or make triple this, now we are labeling manually. I mean, there are the data curators, but that's a lot of effort. We do really need to increase the community of validators or using this type of AI systems. Otherwise, there will be no chances to do it in proper time, because as I mentioned, we want to do this as fast as possible. So that means that by the end of the season, we want all the data already labeled. And that means that we want to do this in nearly almost real time. So what is the idea in long term? To creating these living labs all over the Europe, for example. This can be all over the world, but we try to do so. We need to generate alliances, because these living labs needs to be done locally in local languages. So that means, for example, that we cannot go, I don't know, to the Netherlands. We, as Catalans, even within Spain, we cannot go to the south of Spain, living in the north, because you need this kind of, I would say, touchable. I mean, we go with people to do snorkeling. And we need to create this kind of, community, going with the people sometimes and having these kind of interactions. And this requires, that all what I mentioned in the past of academics, publics, and so on, this needs to create locally. What we can create is the infrastructure that later on help this community each other, for example, on identifying, on knowledge transfer, and so on. And this is our, I would say, long-term goal or mid-term goal, to create such a kind of network of local, living labs that collecting all this data locally, but integrated it in a global, system, that this can create that we can answer then regional or even global questions. And basically is what I wanted to mention. Thank you very much.
time for questions.
Hi.
Lisa?
Very nice presentation. Thank you. So I'm sure you're aware of iNaturalist, right?
Yeah, well, we made a fork of iNaturalist. And people ask why you made a fork of iNaturalist. Basically, because now we are implementing all these kind of new features within the platform that are not in iNaturalist. The problem with iNaturalist is that, you know, it's a very good platform, but it's managed in California. And they decide what they want to do it in California. So whenever we go to a European project and we promise new features, we don't know if they will accept or not. So we needed, you know, a kind of, it's a problem of governance of the platform. Now we can create this network of people and we can decide each other. For example, now we are creating, you know, environmental data. Maybe I naturally say, thanks guys, but we are not interested on implementing the reporting on environmental. And we do need, that's why, you know, we took the original code. This creates another problem if you want. We fork as well the community. Yeah, maybe in the future we can have a way to collaborate.
It's so big.
Yeah.
And people are wanting usually a lot of experts.
Yeah.
Regionally working and uploading thousands of photos and make taxonomy so it's really dedicated people from the bottom up.
Yeah.
And
yeah, but the point is that there is not only iNaturalist. In fact, there are several. So what we are trying to promote is the idea that in many cases, the taxonomists, they do not really care if this picture comes from iNaturalist or artportal observation. So probably we will create a kind of external systems whenever I am, imagine I'm expect on fish or particular taxonomic group, and I am asking there, and then the system can go to the different systems, collect all the data, and me as an expert, I can validate all of them, and then the systems return that. We think, because the problem is not only with iNaturalist is one of the most, but artportal, for example, in the north in Sweden, it's probably in that area more famous than iNaturalist. So probably we're thinking more on the idea of, in some cases, adapting the tools for the taxonomists, and obviously, or trying to facilitate the way that they are exchanged between different platforms.
I had the same kind of question because I think the biggest data providers to GBIT now are eBird, iNaturalist, Artsdatabanken, and the Swedish and Dutch citizen science platforms, and they're research grade. I know you also have research grade photos that are verified.
Yeah, but one of the points, for example, you know, that people sometimes complain is that now GBIF, whenever you use those data, they report it to iNaturalist or artportal or whatever. But what about the people that originally creates the picture? Some people are a little bit disappointed that, hey guys, I put all my effort on reporting all of this and I never receive feedback on that. And I never receive even acknowledge on that. And we think this is really important for, you know, long-term sustainability of the system. So the acknowledge.
That's not true.
Why not?
Because I use iNaturalist all the time and all your information is there, what you provide.
No, but GBIF never reports to you. If you are using iNaturalist, are you receiving, for example?
No, but I don't want to. I mean,
maybe you don't.
Well,
but
why?
What about several NGOs that they want to do it and they want to receive the acknowledgement on all this?
They can get that from GBIF if they want. GBIF do reporting. Yeah.
I don't think so.
But there is a mechanism, I think. Maybe.
Okay, we can talk later.
So you the question.
Yeah, I had a question actually, because I also use iNaturalist and have so many apps on my phone. I have storage, so I don't know where to put them anymore. And it's kind of difficult for a citizen to understand which one is right and which one is wrong. Because not most people are not into science or just want to take a picture of something and help somehow science it, but they don't really know how to do it or which one is more fun. And so having a lot of different ones make it kind of harder. And also, how are you planning to kind of approach different citizens outside of Spain and making it more easy to access and really understanding what they need and how useful it is as well to have pictures of different animals or other sorts of data, not just the AI kind of understanding, but is it good enough? Because like as a citizen, maybe I take pictures of a jellyfish that I've never seen before. I take that one time, the other on the beach, and then never again. Is that like a good solid data or? Like,
Well, I can give you an example. Usually there are like a wide range of collaborations. Some people are really, the ones that maybe just a few that provides thousands of pictures per year, and a lot of people that provides just a very few. But imagine if I have 10 people that are providing 1,000 pictures per year, I got 10,000 per year with this group. But also if I have 20,000 people that provide 3 pictures per year, I have 60,000 for this large community. So we don't need to forget this, I would say, eventual collaborators, because at the end, they can provide a lot of information. Regarding the platforms, I think it's a matter of personal question. If you feel happy with iNaturalist, keep in iNaturalist. If you are happy with Observado, let's do it with Observado.
But as a citizen, I'm talking on behalf of the citizen and not like environment. As a citizen, I don't know which one. It's not about what I like the most, it's about how can I help.
I think there are pros and cons in a sense. So it depends particularly on the local community. Usually the local community at the end ends up with one on another. And I recommend you in that local area, if the majority are using Observado, the best is using Observado. Why? Because you will find several local friends, maybe you can go with them outside and enjoy and so on. So that's my recommendation, but I cannot tell you my feeling, for example, in our area now.
We take Cornelia's question as the last one.
Okay.
No, just a very small comment. I think it's a fantastic example to show what you can actually do, in terms of spatial and temporal coverage in this area that has never been done before. Just on a side note, I mean, it takes a lot of effort to actually get these communities to use it. And I think once you have that, you can expand it also to other areas, and you can also link the data to all the other databases. So I think this is reflecting the huge amount of work that you're putting in your spare time to actually go to the diving clubs and talk to the people and give a feedback to them to actually get these data. And I think we should be a bit more open to use this as a science platform. So
yeah,
great.
You're right. It took 7 years.
That was more common than question. So that's good. We can end on that, and thank all the speakers and Klemens as well. Thank you very much.